Okay, so we're talking about the composition of the plasma membrane, which is really just the cell membrane, right? The membrane that surrounds our cells. It's really important because it controls what can go in and out of the cell. And as you move forward in your understanding of biology, you'll find that controlling what is inside a cell versus outside a cell is crucial to the function of organisms, basically. This control aspect is really important. So to understand that control, we're going to need to understand the components of the cell membrane to be able to see like how it can do that. So the function of the plasma membrane is to define the borders of the cells and also the organelles. So the organelles inside the cell that we'll get to. Remember, when we hear the word organelles, we want to be thinking eukaryotic organisms, right? We had eukaryotes versus prokaryotes. One of our differences was that the eukaryotes have organelles. Regulating what enters and exits the cell. And at the end of this unit, we'll also talk about how the plasma membrane helps cells attach to each other and how the plasma membrane allows the cells to send and receive signals. But for today, we just want to understand what's in the membrane. So when people talk about cell membranes, we often refer to them as fluid mosaics. So we have this thing called the fluid mosaic model. So a mosaic, right, is a bunch of little tiles that make a picture. So we can think about the plasma membrane as a bunch of little tiles, each of which is our different molecules. Okay. So we have 4 different parts of the mosaic that composes our plasma membrane that we want to think about. The first is our phospholipids. If you imagine a cell membrane, you have to imagine a phospholipid bilayer. These other components listed as part of the cell membrane are going to be contained within any cell in different concentrations, different amounts, but we're always going to have like our basic phospholipid bilayer. So phospholipid is a lipid, right? But it has this special quality where it's amphipathic, right? Because it has a polar head that's hydrophilic, right? Attracted to water. And it has a hydrophobic tail. So it has 2 non-polar fatty acid tails, right? This hydrocarbon part, these fatty acid tails are what makes it part of the lipid group, right? All this carbon and hydrogen. Okay. And these are on opposite ends of the molecule. So if we're drawing a phospholipid, we have our hydrophilic head. We got a little glycerol linking together that hydrophilic head with our hydrophobic tails made of fatty acids. Okay. So the phospholipids arrange themselves in bilayers kind of automatically when you drop them in water. That's because we need polar heads that face outward. And when we say outward here, we mean anywhere containing water. A bilayer is 2 layers. So outward in this case means both towards the extracellular environment. So the liquid surrounding the cell, and also towards the cytosol. So that's the internal contents of the cell. So when we say outward and inward, we're talking about relative to the membrane, not relative to the inside and outside of the cell. So polar heads touching the extracellular environment and the cytosol, and I'll talk about the cytosol in the future. And the hydrophobic tails are facing inward, and by inward we mean towards the center of the bilayer, not just the center of the cell. So it's basically we have a polar head, its feet facing inward, and then it's like foot to foot with another phospholipid. So that's the arrangement of our bilayer. You can see it peeled apart kind of here, and then we have in the inset, we have it drawn again, right? We have a polar region on the outside of the cell facing the extracellular environment. We have a big non-polar region in the center of the bilayer, which is like 2 layers of fatty acids, right? Because we have the feet from our first phospholipid, and then the feet from our second phospholipid facing it. And then we have another polar region facing the internal environment of the cell. So that bilayer is really crucial to how our membranes behave. Another note about the fluid part of the fluid mosaic molecule, the fluid mosaic model. When we say that a membrane is fluid, I mean it's like wiggly, right? We're not saying it is water. We're saying that these molecules, right, they're not covalently bonded to each other. They're just kind of hanging out next to each other. So certain types of molecules are going to be able to squeeze between our phospholipids. And in general, our lipids and proteins are going to be able to move relative to each other within the membrane, by which I mean they're going to like move up and down, basically. Okay. Some molecules are getting between those phospholipids, between the components of our fluid mosaic models. But other things aren't going to be able to get through. And that's not because there's covalent bonds blocking them, right? There aren't like bars on this door. The reason they can't get through is because of this nonpolar region. So it's because of these chemical properties of the molecules. So this big nonpolar region in the center of our bilayer, made of these two layers of fatty acid tails, are going to prevent ions from getting through. Now, ions, we remember, are what happens when an ionic compound dissolves in water. It gives us particles that have charges, right? Positive charges are called cations, negative charges called anions. So ions can't get through because even though they might be attracted to this polar head, right, depending on the polarity of that charge, so whether it's positive or negative, they're not going to be able to squeeze between phospholipids because they'd have to pass through this wide nonpolar region. A nonpolar region is hydrophobic and it repels anything that's charged. Same deal with large polar molecules. So large polar, part of the reason is that they're large, but a big thing about why large polar molecules can't get through is that they have the same problem, right? Polar molecules have little mini charges on them. When they reach that nonpolar region of the bilayer, they're going to be repelled by it, okay? So even if they could physically fit in terms of like how big they are, they wouldn't be able to get through this region because of their chemical properties. Okay. So this is part of why we can also refer to the membrane as selectively permeable. So selectively permeable means it's going to allow some things to go through it, and it's going to block other things from going through. This is important because we want essential molecules to enter. We want the middle parts of our various chemical reaction pathways to stay inside. We don't want them to float out into the outside environment before we're done with them. So that's what this means by metabolic intermediates remain, right? So things we need in order to continue a chemical reaction, we need them to stay inside the cell. But we want stuff to leave the cell, right? We might want signals to leave, but we're also going to want our waste products to exit the cell. So we're going to need to be able to control what goes in and out. The first step of that is this membrane that blocks ions and large polar molecules from going through, but we're going to allow small nonpolar things to get through because they can squeeze through this nonpolar layer. So our second component of our plasma membrane is cholesterol. So cholesterol is a steroid, which means it's a lipid, which means it's a hydrocarbon. It's found in animal cells, but we're mainly thinking about animal cells, which is why it's on our list. So it wouldn't be in a plant. One of the effects of cholesterol is that it changes this membrane fluidity. Most membranes, it makes firmer, but it also sometimes acts as a buffer. So it keeps this sort of jiggliness of the membrane within a certain range, because we don't want the cell membranes to jiggle too much because we don't want them to jiggle apart. But we also don't want a super rigid cell membrane in an animal, right? Because then we wouldn't be able to get things into the cell, because part of the way we're getting important nutrients into the cell is by allowing them to squeeze through the membrane. There are other factors that affect fluidity, but honestly, we're not really going to talk about those in these class. But in case you're curious, temperature and phospholipid type would make a difference. And when we say phospholipid type, that's basically like the shape of those fatty acid tails. We talked about different types of fatty acids, so you can kind of imagine in your mind how that can make a difference. Okay. Our third component, which we're going to talk a lot about in the future, we're introducing here, are the proteins. So proteins are going to be available in varying amounts in different types of cells and at different time points. But they're really important to how we control what goes in and out of the cell. So they're going to be crucial for what we call transport, which is going to be our first topic next week, which is going to be the controlled movement of things across our membranes. They're also going to act as receptors, so they're going to receive signals. They're going to act as enzymes, so they're partially going to control reactions that go inside the cell. And we'll also see that they're part of cell junctions, which is to say how cells attach to each other. Now we have two types. Integral proteins go through the membrane, and here the key part we want to see is that they go into this nonpolar layer. And peripheral proteins are just going to kind of sit on one side. So that's like this green one, just kind of sitting on top of our phospholipids over here. So those don't reach into the nonpolar portion if it's a peripheral protein.
Can I ask a question real quick?
Yeah, what's up?
So integral proteins, do they have to go all the way through the membrane, or if they're just invading the nonpolar part at all, they're considered an integral?
They're considered integral proteins if they go into the nonpolar part. Really, the ones we talk about are transmembrane integral proteins, so they do go all the way across. But yeah, it's the integral protein means that they go into the nonpolar region. So they would look like this. back to those integral proteins, right? So integral proteins are able to go through the membrane, go into that nonpolar region, because they're going to have at least one region that's hydrophilic, so attracted to water, either outside or inside the cell. And they're going to have a region that is hydrophobic, so a region that's nonpolar and able to insert itself in this nonpolar region, right? So here we see this part of the protein that's touching those fatty acid tails is going to have to be exposing nonpolar R groups. So as you're working through your problem set, if this is kind of confusing, go back and look a little bit at our levels of folding in proteins and different protein properties in order to understand exactly how you might end up with this. But basically, it's those different side chains on our amino acids, depending how the protein folds, will be on the outside of the protein. And so they can give different parts of the protein different chemical properties. Okay. Our final group are carbohydrates. So carbohydrates basically can get tacked on to our different molecules in our cell membrane on the outside. So they're mainly focused on like cell-to-cell recognition and attachment, so your body knows what type of cell we're looking at. And they're either going to get bound to one of these proteins, right? So here we see a glycoprotein. Glyco is telling us basically that this is a sugar. This is a prefix that kind of indicates we're talking about a carb. And protein, because it's attached to a protein. Or we have glycolipids. So if we have a glycolipid, we might have we got a green one over here. We also have one over here. We can see that we have this little chain attached to one of our phospholipids, right? So that's where our carbohydrates come into play in terms of our cell membranes.